would suggest that, in fact, certain subspecies of HDL can play a key role in host defense mechanisms against invading pathogens. So this is kind of illustrating what we think is a paradigm shift in thinking about HDL. We have the old notion, based on the nutrient delivery hypothesis, that the major function of LDL and VLDL is to deliver these nutrient-rich triglycerides from the liver to the adipose tissue. And in this scenario, HDL is kind of a passive player that just accepts cholesterol from macrophages and takes it back to the liver for its excretion. In our model, we have a very different way of thinking about this. We think that HDL is inextricably linked to macrophage activation and host defense mechanisms. And we don't think that this is just kind of a passive process where cholesterol comes off the surface of the cell and adsorbs onto HDL. We think that it's a dialogue between the HDL and the macrophages, and that this in turn plays a very important role in controlling inflammation and perhaps in controlling infection in humans. And there's already strong evidence from many other investigators that proteins can jump from macrophages onto HDL, consistent with this model. And so what we would speculate is that proteins and lipids are being exchanged between HDL and macrophages, and this is playing a key role in regulating how macrophages respond to inflammation and infection. So another interesting component of this hypothesis that the proteins in HDL can be very specialized and have many different functions is that they might also be playing a role in heart disease. So this is coming back to the idea that there could be dysfunctional HDL in humans. And so we thought it would be interesting to look at the protein composition of HDL isolated from humans with established heart disease and control subjects. And again, we used mass spectrometry to do an unbiased analysis of the protein content. So this is a fairly complicated experiment, and I don't have time to go through all the details here, but this is showing you all the proteins that we detect in HDL. And on the bottom is an axis for whether or not the proteins are enriched in one group of subjects. So if a protein has a value of 1 on the bottom axis, it means it's only found in people with heart disease. If it has a -1 value at the top of the graph, it means it's only found in people who are healthy. And if it has a value of 0, it means it's equally abundant in people with and without heart disease. And what you can see is that there are 5 proteins that appear to be enriched in the HDL of people with heart disease. And interestingly, 4 of these 5 proteins are secreted by macrophage foam cells. And so this is a relatively small study. Mass spectrometry is not an easy thing to do in a high throughput form. But we're very intrigued by the idea that altered protein composition in HDL may be a marker and perhaps a mediator of heart disease in humans. So to summarize then, we think that normally HDL is cardioprotective by at least 2 distinct pathways. One involves the removal of cholesterol by ABCA1 and other pathways. This decreases macrophage sterol content and makes them less angry. We also think that proteins in HDL are regulating inflammatory pathways in macrophages. And we have very strong evidence using array analysis of macrophages that this hypothesis is correct. We also like the idea that there could be dysfunctional forms of HDL in humans. And I've outlined some of our work on oxidative damage of ApoA1, and the notion would be there that by damaging specific amino acid residues in ApoA1, we block the ability of A1 to remove cholesterol from macrophages, and this increases their sterol content, their cholesterol content, and makes them angry. We also believe that altered protein, and I should mention perhaps lipid composition of HDL, this is not something we've looked at but we're very interested in, also can play a role in regulating inflammation. And we would speculate that alterations in the protein or lipid content of HDL may also be playing a pathogenic role in atherosclerosis. So where do we need to go with this? What are the big questions? Well, one question that fascinates us that may seem trivial to many people is just what is HDL? The way that we traditionally isolate HDL is to put it in a very concentrated salt solution and to spin it at very high centrifugal force for 48 to 72 hours. And it's long been known that this dramatically alters the protein content of HDL. And I've already mentioned this idea that what we think HDL actually is a whole series of specific protein-protein complexes. And so what we speculate happens during this ultracentrifugation step is analogous to having a whole family of tinker toy type constructions or specific macromolecular complexes that are essentially randomly dissociated. A third of the proteins are thrown away, and then you just add them back together into some kind of scrambled mixture. So we're very interested in using other biochemical techniques to isolate HDL in ways that don't destroy the particles and to go back and ask about some of these fundamental questions about macrophage biology. We're extremely interested in the idea that HDL is talking to macrophages and regulating inflammatory pathways. And we don't think that this is relevant only to atherosclerosis. Many different diseases involve angry macrophages. Alzheimer's disease is characterized by angry macrophages. Many forms of inflammatory bowel disease are characterized by macrophages. Lung disorders, macrophages. In fact, we've been looking at the protein composition of macrophages, and the proteins that we find secreted by foam cells are also highly abundant in Alzheimer's disease plaques. So we're very interested in the idea that HDL is regulating novel inflammatory pathways in macrophages. And finally, we think that by looking more carefully at the protein composition of HDL, we might be able to come up with a better way of diagnosing people at risk for heart disease. So although everyone gets their LDL and their HDL measured on a routine basis to try and assess cardiovascular risk, it turns out that they're not very strong predictors of risk in individual people. In fact, you can do almost as good by flipping a coin as you can by measuring an HDL and an LDL in an individual. And so we're very intrigued by this idea that by looking more carefully at specific protein components of HDL, we might be able to come up with tests that tell us a lot more about who's at risk for heart disease and whether or not our therapies are working. I'll just mention, for example, that our best therapy for preventing heart disease, statin therapy, which is one of the biggest successes in medicine today, only prevents a third of heart attacks. And we have no idea how to identify the people that are benefiting, and we also don't know whether or not changing HDL could further improve those results. And so we think that this lipoprotein-protein complex that's long been thought to just be playing a passive role in transporting nutrients actually may have far more fascinating properties, and that there's a long way to go in terms of understanding what's happening with these particles. So with that, I'm going to end my talk, and I'd be happy to take any questions, assuming there are any questions. Let me just thank the people